In this video, we will discuss about the nucleic acid therapeutic development, how this nucleic acid or mRNA based technologies has been developed. So it all starts with the DNA isolations by DNA isolation by this Friedrich Meisser in the form of chromatin, and then it begins that in 1977 the DNA sequencing technology has been developed, and in 1983 the PCR technology has been developed, and it comes to the point where in 2009 first mRNA based cancer vaccine trial has been done, and in 2020, as we all are familiar with, this mRNA based vaccine for COVID-19 has been approved for emergency use. So this is in brief how this mRNA technology development occurs, and in the panel A of the right hand side of the figure shows different kind of RNAs. So in brief, they are categorized into two different kind of RNAs. First one is long chain RNAs, and another one is short chain RNAs. Short chain RNAs are mainly used to suppress or affect this gene, suppressed or down regulate the genes, we can say that, or modulate the genes, whereas long chain uh, RNAs have some different functions. And similarly, this DNA has been divided into different parts. DNA can be either knocked down, silenced, and these things can be done either through some exogenous reagent or by editing the DNA using different techniques such as CRISPR-Cas knockout. And in the panel B of the right hand side figure, panel B of the right hand side figure shows some of the cellular based approach that are currently being used. So for this mRNA therapeutics, one approach is that it can directly be introduced exogenously, or that is shown in this, that is represented by this therapeutic protein productions. Or what it can do is it can in the case where exogenous mRNA therapeutic approach, mRNA has been exogenously introduced into the cells or transduced into the cells, and then it interacts with the ribosome for the therapeutic protein productions that can further does it functions to suppress the proteins that is causing the disease. The other approach is the prevention of pathogenic proteins through this ASO. So this ASO will bind to the pathogenic proteins, and thereby it will disrupt the ribosome to translate this mRNA into the proteins. The other things is this prevention of pathogenic proteins by ASO recruiting RNAs. So in this case, ASO is bind to the pathogenic proteins, and then it will recruit RNase S. So such that this RNase S will degrade this mRNA and thereby inhibit the pathogenic productions. The other approach is SSO based approach, where this in this case the mRNA will interfere the splicing mechanism of the cells in such a way that these pathogenic proteins productions will be disrupted. And next one is this DNA editing nucleic acid. In this DNA editing enzymes such as CRISPR-Cas9 systems can be introduced into the cells, then it will splice or edit the pathogenic proteins sequence so that it's functional, so that the functional pathogenic proteins will not be produced. Next one is this double-stranded endogenously produced DNA. So in this case, the double-stranded RNA, not DNA, RNAs. Double-stranded RNAs is uh, transduced into the cells exogenously, and it will uh, interact with the pathogenic proteins, not 100% complementary, but to some extent, as it will form a hairpin that we can see in this figure. And what it will does is this: it will interact with the proteins and thereby prevent these proteins to perform its regular functions. And the other things is the disassembly of the protein translation machineries. So this uh, exogenously complementary double-stranded RNAs. So what it will does is will interact with these pathogenic proteins and will prevent the pathogenic proteins formations or inhibit the pathogenic proteins translations. And in the case of 100% complementary double-stranded RNAs, then in that case what will happen, that it will recruit this RISC helicase unveiled double-stranded it will recruit this RISC that will degrade these complementary mRNAs and prevent the pathogenic formations. So these approaches, the exogenous complementary double-stranded RNAs, are somewhat combined to each other or related to each other, we can say that. So these are some of the approaches that we have discussed in the nucleic acid therapeutic developments that has occurred with time period, so that we have reached to a stage where we can use those approaches for the diagnosis of the diseases at cellular level. That's all for this video. If you need to understand more about this developments in detail, you can go through the reference that has been provided in the description. Thank you for your time.